All right, recording in progress. Welcome everyone to the Director's Choice seminar today. It's just gone 1 o'clock. My name is Dr. Adrian Wiegmans. I will be presenting with my PhD student today, Kate Ivanova. I joined the Cancer and Aging Research Program early last year as part of QUT. Under Derek Richard, and today I'll be presenting some of the work that we've been doing over the last 2 years. This is deciphering the links between chemo-resistance and metastasis in breast cancer. Let's see if I've got I can see that there. Here we go. I have a pointer. Now, as you can see, on the right, I have a schematic of the metastatic cascade there, and there's actually 2 points that we're really interested in. Firstly, initially, the capability of cells to escape therapy, so as they become chemo-resistant, and looking at those mechanisms and phenotypes within those cells, and whether those phenotypes help those cells survive in the circulation. Our overall goal is actually to create some molecular diagnostics that help to guide therapeutic choices. We're doing this for the triple-negative breast cancer subtype. This is actually one of the most aggressive subtypes, and under the current clinical use, they still use chemotherapy to treat these cancers. The first half of this talk has been published recently in NAR Cancer. And what I'll be talking about today is I'll go through a little bit of the background, how we created these chemo-resistant triple-negative breast cancer cell lines, how we elucidated the mechanism of resistance, some of the current work that I'm looking at with aspects to liquid biopsy and biomarkers, and then move on to Kate's PhD project, which she's just started, is looking at the non-adherent cell cultures. So firstly, let's jump into the background. As you may or may not know, the Cancer and Aging Research Program focuses on the DNA damage response. And as you can see here, it's quite a complex set of pathways that feed into other pathways. And I thought I'd just really dumb this down a whole lot, make it nice and easy. So there's two main pathways that deal with double-strand breaks. One is the homologous recombination pathway, which is high fidelity, and we have a lower fidelity non-homologous end joining. And these actually result in repair of DNA breaks, whether they come from endogenous. From in triple negative, we have replication stress that occurs, or whether it happens from chemotherapy-induced breaks. And actually, in our project, we see that there's actually a switch between non-homologous end joining towards homologous recombination during the acquisition of chemo-resistance. So the chemo-resistant cell lines that we created, we treated them with the frontline combination therapy of doxorubicin and docetaxel, and we treated them with escalating doses over the course of about 6 to 8 months, and they adapted to growing in these high levels of these drugs. But first I wanted to have a look at, is triple negative really guided by the DNA damage response, and how is that an important part within that subtype? So we went to clinical trial data and had a look at the gene expression profiling and also the patient outcome. And as we see that when you're treated with chemotherapy, the triple negative breast cancer subtype does a lot more, well, the patients do a lot more poorly for their distance relapse-free survival versus all the other subtypes. We had to look into the gene expression profiling of these patient samples. And it's a little bit hard to read there, but I'll say that we did a gene ontology pathway analysis. And of the top 10 pathways that were induced, 4 of them are related to DNA repair functions. So firstly, we had a macroscopic view of our cell lines, and we just wanted to see what was going on. So we're looking at the chromosome counts, and actually 2 of the cell lines that acquired high levels of resistance to the drugs that we're treating them with actually showed losses of chromosomes on a large scale, whereas the other 2 that didn't acquire these high levels of resistance did not. So we actually find that there's a correlation between loss of chromosome number and the achieving of chemo resistance. So this led us to the hypothesis that it's probably non-homologous end joining, so there's less fidelity and we're getting loss of chromosomes. So looking into the mechanism of resistance, we treated our breast cancer cell lines that match sensitive and resistant with key small molecule inhibitors that inhibit these particular key proteins within the pathways. And if we have a look at the pie graph down the bottom, as we see in our sensitive cell line, there is actually a nice distribution between the use of non-homologous end joining and homologous recombination, as we would expect throughout the cell cycle, and they're using both of these pathways simultaneously and as they are required. However, when we look at the resistant cells, we actually see that there's a large reliance upon homologous recombination. So this was unexpected. We thought it might be non-homologous end joining. So we created this, we saw this scenario with creating double-strand breaks using a very classical scenario of gamma irradiation. And we're able to replicate these results in the presence of our chemotherapy combination. We also validated these results using immunofluorescence. So immunofluorescence assays is the tagging of some of these complexes within the nucleus using fluorescence, and we can track that over time. So it's the ability to really repair these breaks over the span of a day. And we see in our sensitive line, if we look not primarily at the foci, but we look at the graphs. There's my pointer, here we go. RAD51 is the key protein involved in homologous recombination. We see there's an induction to about 30 foci per cell, and then we get a nice resolution. And in comparison, once again, up to around 30 foci. So we can see, again, this is a complementary between these two different pathways. And I didn't mention that, sorry, this graph here is a representation of phosphorylated DNA PK, which is the key protein involved in non-homologous end joining. However, when we look at our resistant cell lines, we see, again, the trend is towards a high involvement of homologous recombination and dependence on that type of repair, and not so much non-homologous end joining. So fortuitously, I've been working on homologous recombination for a while, and we actually created a few years ago a small molecule inhibitor against the main protein involved in homologous recombination called RAD51. So we utilized our RAD51 inhibitor, and we're able to actually resensitize. So we see with chemotherapy, we have a nice resistance here, and we can sensitize these resistant cells, or even more sensitive than the sensitive cells, with a RAD51 inhibitor. And it's also available on the market, a RAD51 stabilizer. So just to show that when we add the stabilizer in, we actually create increased chemo-resistance. Now, what was interesting is that when we added our RAD51 inhibitor in the presence of chemotherapy, we were actually able to achieve very similar levels of cell death between sensitive and resistant lines. And so we looked at the gene expression profiling within these two cell lines. And again, we see in the sensitive cell lines, we see a combination of non-homologous end joining and homologous recombination contributing to the repair. Whereas in the resistant cell line, we only see the contribution from homologous recombination. So even though the cells are trying to repair, they're both utilizing it in different pathways. So here we come to the model that we propose and have published. So there's actually a stress on non-homologous end joining that we see the loss of chromosomes and this very low fidelity repair until there's so much stress on that particular pathway that it actually shuts down and then switches over to homologous recombination. So I'll just move quickly on to the current work. So I wanted to see and look at whether we could utilize this mechanism as a chemo-resistance biomarker, and in particular a liquid biopsy biomarker. So in triple negative, you really want to monitor the patients and you want it to be as non-invasive as possible. So the ability to take a blood sample or routinely take blood samples and have a look at markers within the blood to have a look at whether the chemo-resistance is arising or not is actually one of the clinical scenarios that they really want. So looking at this clinical trial data, this is actually a really nice clinical trial that was set up using the same drugs that we use, the docetaxel and doxorubicin. And they had matched samples of untreated and post-chemotherapy neoadjuvant treated patients, and we could look at gene expression. And what was interesting, they see the same phenomenon in their patients. So it's this key protein involved, or this is, at this point, this is a gene expression for non-homologous indel. And we see this pressure on this pathway, and there's actually a down-regulation as the cells try and evade that type of response. So I look at the rest of our key interesting genes related to DNA repair, and we can see there's, what's this, 5 genes that are induced with chemotherapy. XPC, the first one here, is actually really a backup of a backup, really. It's involved in, base excision repair. And so it's the cells trying to use everything in their arsenal to repair the DNA. Now, what was interesting that we thought was this kinase that comes in second, c-Abl. So it's a signaling kinase. It's involved in a lot of different pathways. So I thought that was quite interesting. So we did string analysis, and knowing that homologous recombination repair is important, we put in all this gene expression profile data and did a string analysis and found this. Again, we find homologous recombination repair node, but there's also a signaling node as well that relates. And again, this is important for c-Abl. So looking at c-Abl in more detail, we have ATM kinase that is an important DNA repair kinase upstream, and it's one of the universal signaling kinases for DNA repair. And when we plot all the patients from the clinical trial for untreated, we are able to look at the correlation between the expression of ATM and c-Abl. There's no real correlation there until the patients are treated with chemotherapy, and we see an upregulation of c-Abl. And this actually translates really nicely to patient outcome. So when patients have low c-Abl expression levels, they actually do quite well on chemotherapy. However, if they have high levels of c-Abl, chemotherapy doesn't assist them at all. So moving forward, I had a look at the literature and thought, well, what regulates c-Abl expression? And looking into different transcription factors and things of that nature, and it really didn't show up. There wasn't a correlation between the transcriptional regulation. So I was wondering, all right, what other levels can this be regulated at? And microRNA-7 is known to be involved in chemo-resistance, and it was shown in some breast cancer cell lines to be regulating c-Abl, and that's great. But also I found that circular RNA, this new molecule of intron splicing, it's a fault with intron splicing, and it creates these circular RNAs. Circular RNAs mop up mirrors. So what the theory was, so this was shown in leukemia, that this particular chemo-resistance associated circular RNA was mopping up mir7. So I thought, well, this might be what the mechanism is. So how you amplify a circular RNA is that you can do it with a single primer. You do something called a rolling circle PCR, and you do one primer and it just rolls and rolls and rolls and rolls until you get a nice length of product. So we look at the conditioned media of our matched sensitive and resistant cell lines, and as you can see, where's the pointer again? Can't see it. Anyway, in orange, we can see our sensitive lines didn't amplify this circular RNA, whereas we can actually detect it in our conditioned media of our resistant triple-negative breast cancer cell lines. So this is great. So we've got this resistance mechanism, maybe, the regulating c-Abl, but I had to prove it a little bit more. So I did some qPCR analysis using mir7. So it's mir7 inhibitors and mir7 mimics available. So when we add these to the cells, we can actually modulate the levels of c-Abl, showing that it was really good. But how does this relate to anything important? We can also modulate the levels of chemo-resistance in the cells with this mir7 mimic or inhibitors as well. Now, this is where I find myself at the current time. I've got a little bit more analysis to do as far as looking at these particular molecules in patient samples. What's interesting as a clinical biomarker is that circular RNA is very stable in the serum. Because it's circular, it can't be attacked by RNases. So it should be able to be detected. So this is the next step that I'll be looking into. So I'd like to pass over now to our PhD student, Kate. And we'll actually suspend, I read that suspension. We'll suspend questions till the end. We'll take some questions at the end. We'll take them through Q&A on Zoom. And I'll hand over to Kate now. Thank you.
Thank you very much. Hello, I'm Kate, and I've been a student of Adrian's for quite a while. However, I only started my PhD a couple of months ago. So in this part of the presentation, I will focus mainly on our ideas for the project, ultimate goals, and I will show a little bit of experimental data that I have accumulated at the moment. So we've had this idea that DNA repair, faulty DNA repair pathways fit into chemo-resistance, which eventually leads to metastases. So we wanted to show this link between chemo-resistance and metastases and DNA damage repair. We also wanted to find a way to bring our findings into the clinic. And this is when we thought liquid biopsy sounds elegant, has a huge analytical potential. However, currently it has very limited use. So we wanted to fit into this idea. And this is how we came up with suspension cultures, having already established matched isogenic cell lines modeling acquired chemo-resistance in breast cancer. We decided to adapt our cell line pairs to non-adherent conditions and see what happens. And there's a fair bit of literature out there on similar studies which use this, a similar concept to ours, namely putting adherent cells into suspension. And what they seem to show is that cell lines, cells, as soon as they're exposed to low adherent or non-adherent conditions, they become primed for metastasis. They become more efficient at spreading. They become more stress resistant. They modulate their metabolism in order to adapt to the changing conditions. And this last study, whose screenshot you can see on the screen, particularly resonated with me because we've been looking at DNA damage repair in our chemo-resistant cells, and there we go, we see that as soon as adherent cells are exposed to circulating conditions, they pre-activate their DNA damage repair pathways and therefore become more resistant to chemotherapeutic drugs. So they upregulate their innate resistance to therapies. As Adrian has already mentioned, we had breast cancer cell lines, which were isogenic, chemo-resistant sensitive breast cancer cell lines, isogenically matched, which we have been growing in adherent conditions. And as the main idea of my project, we subjected them to low adherent conditions and generated over time suspension cultures, stably proliferating suspension cultures. The idea behind this was to investigate which comes first, chemo-resistance and then metastases, or so is it chicken or the egg? Which event occurs first? And also, as previous studies have shown, suspension state enhances drug resistance in sensitive cells. So we wanted to see what would happen to cells that are already resistant, that while they have been grown in adherent conditions, they have developed this chemo-resistance. So what will happen to them when they are subjected to circulation, to suspension conditions? Will they become even more resistant? As we found out, chemo-resistant cells have a harder time adapting to suspension culture. We are yet to investigate as to why exactly that happens. But now we'll show you a couple of images that I took of the cells that I've generated over the last 6 months. So we started out with adherent MGM-B231s, and in the later slide, it will be B259s. both are triple negative breast cancer cell lines of different molecular subtype. As you might have heard, triple negative is a molecularly very diverse cancer. And using cell lines of different molecular makeup might provide more insights into mechanisms of chemo-resistance in this particular setting. So as you can see in the image on the right, our suspension cells tend to grow in clusters. But upon slight agitation, sensitive cells dissociate from one another and they continue growing on their own, forming their own respective clusters. However, this does not seem to be the case with our chemo-resistant cultures. They really like to stick together. They form very tightly packed clusters, and you really have to work very hard in order to dissociate them from one another. You have to trypsinize them for extended periods of time. You have to PET them, sometimes in vortex, and even then, there's no guarantee that you will end up with single cells. So this raises obvious experimental questions. What about proliferation? How do these cells grow? Because supposedly cells at the very center of the cluster will be contact inhibited by other cells. And does it affect drug delivery? Because cells right in the middle of the cluster will be shielded from any drugs that we might put on our cells by the cells on the outer layers of that same cluster. Yes, as I mentioned, sensitive cells dissociate from each other very easily. All you need is to shake the flask a little bit, and what you started with as clusters on the left-hand side, you end up with smaller clusters or even single cells, as you can see on the right-hand side. And resistant cells tend to grow in very, very tightly packed clusters, which are not only hard to trypsinize and dissociate, but they're very hard to image because you have to scroll through multiple layers and they never come in true focus. So having established our suspension cell lines, we decided to pretty much repeat all of our experiments that we did for our previous paper, looking at chemo-resistant sensitive cells. And surprisingly, what we found is that somehow our super-resistant cells became more sensitive to higher concentrations of our combination chemotherapy. It is a very interesting observation. The literature does say that sensitive cells supposedly become, as I mentioned earlier, they supposedly become more resistant to various stresses, including DNA damaging agents, as they have pre-activated their DNA repair pathway. So that explains why we see good viability of sensitive cells at high concentrations of chemotherapy, but that does not explain why our resistant cells become resensitized. We were still awaiting, due to lockdown, we were unable to perform cell cycle assay, which would confirm once and for all why we were seeing this effect. But my theory is that resistant cells reduce their proliferation rates once they're exposed to chemotherapy, and instead of undergoing apoptosis or any other form of cell death, they just reduce their proliferation rates. Why do I believe so? Well, it's because before we started with these experiments, we routinely reselected our chemo-resistant cells by subjecting them to a week-long incubation with high doses of doxorubicin docetaxel. So I can be fairly certain that the cells that we're working with are truly chemo-resistant. And as I said, we are awaiting our results from cell cycle assay, which we were unable to perform leading up to this day due to the recent lockdown. Switching from adherent culture to suspension has had its own challenges. We had to rethink our methodology because the original idea, as I said, was to repeat all of our experiments from the previous paper using the new cell lines. But due to the fact that now we're dealing with floating cells, suspension cells, we had to change we had to modify our experimental conditions. For example, as Adrian showed you in his previous slides, we examined DNA repair dynamics using immunofluorescence, and that was very easily done in a 96-well format using our wonderful in-cell that we have in the lab. However, with suspension cells, it will be tricky, and we're still figuring out how exactly we are going to approach this question, because we can either do flow cytometry, but then again, we would have to make sure that we are really dealing with single cells, and resistant cells tend to form clusters, or we could do immunofluorescence, but in a more conventional way. That will require more processing, so we'll need to figure out a way to reconcile that. So my project, so far I've only been talking about the lab aspects of my project, which and the work for this part of my project is currently ongoing. We are aiming to characterize DNA damage DNA damage repair efficiency in our suspension cells, whether they become better at repairing double-strand breaks, or as in the case with resistant cells, they so we are planning on characterizing DNA repair dynamics in our suspension cultures. We are also awaiting results from our RNA-seq, and this will allow us to formulate gene expression profiles and compare our sensitive versus chemo-resistant, as well as adherent versus suspension cultures. However, this is all purely lab-based, and we are truly hoping that eventually, sooner rather than later, my project will have a more clinical component to it, because originally it started as a very translatable idea. It's something that we can readily bring into the clinic to make patient quality of life better. So we are currently in contact with the several clinicians from the PA hospital, and we've just submitted a grant application, so we are expecting a positive outcome. So hopefully we'll be able to recruit actual triple-negative breast cancer patients from whom we will collect blood samples and isolate circular RNAs for Adrian's part of the project, and I will focus on circulating tumour cells. Coincidentally, maybe not really, I have already been involved on a different project relating to circulating tumour cells. And thanks to Dr. Arutha Kulasinger, we now have an established workflow how to isolate circulating tumour cells from patient blood samples. It involves a microfluidics chip, which has 2 channels, and one of the channel will contain circulating tumour cells, which will then be spun down onto cytospin slides, as you can probably see on the screen, because I don't know, you can, okay. And we'll use those slides to do immunofluorescence. I also would like to attempt CDC cultures. It is a very challenging feat, but I'm very hopeful. And if we do succeed, it will be quite interesting to look into that aspect as well, because having CDC cultures growing for a longer period of time will provide us with more material to work with, and as a result, more data. And just thought I would show you some nice images from my recent CDC isolation. So what you can see here in green is a very big CDC cluster that we isolated from lung cancer patient blood. So the reason how we know that it's a true circulating tumour cell cluster is because we used a pan-CK marker, pan-cytokeratin, which is a marker of cells of epithelial origins, and cells that are of tumour origin, they stain really well for that particular antibody. Sorry, I just got distracted. Yes, so these cells are positive for pan-CK and are negative for immune cell markers, which are CD45 and CD66B, which is a marker for neutrophils. But it is very interesting to see that, as a lot of literature shows, Tumour cells in circulation tend to form clusters not only with each other, but with immune cells, which allows them to metastasize better, evade immune system. And in these images, we see it really nicely how tumour cells travel together with immune cells. And this is it. If you have any questions.
I get the start time now.
Yeah.
Oops.
Exactly half an hour. Oh no, what happened?
What's happened here? We did our AV people.
Do you need to go back? Can you?
Wait a minute. If I stop sharing all that.
No, you have to continue.
Are you trying to end the meeting?
No, I'm trying to find the here we go. Questions, chats.
And Adrian, if you can stop sharing.
So if you mouse over the green part, it'll pop down the extra options.
All right. Oops. Oh, here we go. Yep. Oh, this screen is very bright. Oh, great. All right.
And Adrian, if you could stop
We've got a lot. All right. Let's start at the top. From Nick. Chemo makes chemo worse than no therapy.
Can you read it out?
All right. High c-able makes chemo worse than no therapy. Possible contraindication. High makes chemo. Yeah, that's good. Well, yeah, that's a good point. I won't bring back the graphs. It doesn't actually dip below the low graph. So it's not actually worse per se. Kate, proliferation and drug. To Kate, proliferation and drug accessibility will depend on multiple factors, not only clustering, cluster size, ECM. Yep, excellent.
Yes, but at this stage, we're not co-culturing, so we're only working with one type of cells. And also another factor that it depends on is whether or not we have an upregulation of P-glycoprotein pumps, and that has been excluded with these two particular sets of cell lines, as we did try to derive chemo-resistant cell lines using other TNBC cells, but they did upregulate PGP, and the ones that we're using for this project, they do not have upregulated drug efflux.
Does anoicus contribute to overcoming the resistance? I think it has a role, actually. That's true. I've seen a differential regulation of the gene FAC at the moment, so I'll be looking into that because that's quite interesting. Rick, sorry, I have another meeting. All right, Rick. Can you clonally expand your CTCs in vitro or use a xenograft mouse model? From the history, patient-derived CTC cultures are very hard to establish. We've been trying to track down through international collaborations any breast cancer CTC models, and they can keep them alive using various cocktails of cytokines, et cetera, but only for about a week or so. Can we use in vivo xenograft? Yeah, we can use xenograft mouse models. We have a collaboration, Salt Lake City.
Oh, yes.
They do really good xenograft mouse models. Great talk. Here we go. Pascal, thanks, Pascal. Great talk. In drug-resistant cell lines showing multiple chromosome losses. Were there specific chromosomes that were currently lost? Oh, sorry, we didn't. Yeah, I didn't get down to that level. Metaphase spreads, we could do something a bit more. We did SNP analysis, SNP arrays, so we could probably have a look at that, copy number variation and match it all up. No, didn't look at that level. Stop sharing your screen. All right, from Teresa, would regulating cerebral within patients result in adverse effects? There is one clinical trial underway with c-able inhibitor. We used c-able on our samples. Actually, didn't really do much at all. I think it's because it's at the intersection of so many different signaling cascades that it can be bypassed really easily. So as far as a biomarker, good. Target, no good. There we go. Any more questions quickly through, anyone? Q&A, no, nothing Q&A. We're very chat orientated. That's fine. Thanks everyone for your comments. Please email anything through if you think of something later on. That's all right. So thanks everyone, and good luck from wherever you are zooming in from, lounge room, office, etc. All right. Thanks guys. And let's end.